valuable time. Over to you, sir, now. I'm really sorry to interrupt you, sir. Respected sir, please just put on your mic, sir. Thank you.
Yeah, can you hear me now?
Yes, sir.
Yes, sir.
It's
Yeah.
completely audible. Thank you, sir.
Thank you very much, Dr. Vikas, for your kind introduction.
Thank
And
you, sir.
it's a pleasure for me to interact with the young students, especially because I would like them to take this field up as a career option for them. And we welcome all your students who want to come here and do internships or some kind of fellowships in our company. And we wish your students all the very best. So just a brief history of Virchow. Virchow Biotech was founded in 2001, and last year our revenues have crossed over 700 crore rupees.
No, great.
And we deal with a, a lot of different biological products, including biosimilars, recombinants, blood products, vaccines, traditional biological products such as wound healing products and other enzymes. So we have a rich history of dealing with biological products in our company. So why are we here today? We are here today to discuss the new field, not new field, but new to some of your students, the new field of biosimilars. So what is uh, the outline of my talk will be just an overview of the biologics industry and an approach to the biosimilar product development, how you would approach developing a new product, and some case studies that we have. Uh, and talking about documentation, why documentation is very critical. So just a glance of the market size in last year, if you look at this screen, you can see that adalimumab, which is also known by the brand name of Humira, generated sales of over $20 billion. And other traditional maps such as rituximab, denosumab, trastuzumab, they all had over uh, $2 billion in annual sales. So this represents a huge need in the market for these biological products because they are used for life-saving uh, purposes. And the whole idea is that since some of these products don't fall under intellectual property, we can duplicate or make a similar product and use them for meeting the unmet needs in our population. So this is just to show you the different uh, FDA approved monoclonal antibodies. And this list is very long, and there are different types of therapeutic indications for these products, going from uh, cancer treatment to psoriasis, multiple sclerosis, and other different indications. These are some of the products that we currently are working on. Actually, one of our products, rituximab, is in late stage clinical trials, and the clinical trials will be finishing soon. And then we will hope to approach the regulatory body in India for marketing approval. So in the US, the biosimilar market is also picked up, which is initially it was called as a generics, which were the small molecules. Then they realized that India is providing a lot of competition for the small molecular space in the US. So they decided to uh, cut out the word generic from biological products, saying that you cannot have a generic in biological products. And they called it as a biosimilar in a way to restrict the marketing of these products by other companies and to protect their market share. But actually, if you look at it, since this is a biological product, even the innovators' products are not similar to each other. If you look at multiple lots of the product there, they will all be varying from each other. So in that sense, even they are biosimilar players. So these are just some kind of biosimilars market share in the world. out of which monoclonal antibodies form a big chunk of it. Then GCSF, which is the granulocyte colony stimulating factor, uh, is also a big part. Erythropoietin and analogs, insulin and other analogs, growth hormone. So these are all the different areas of growth. So what is the life cycle of a biosimilar? First, you do research and development. You make the product that is meeting or matching your specifications. Then you check for its quality, which means efficacy and safety. Then you check for manufacturability. And then you look at the scale up of the process. Again, research and development, manufacturing, and then regulatory aspects. So typical reference biological product development undergoes the following stages: discovery, development, preclinical, then phase 1, phase 2, phase 3 studies. Whereas for biosimilars, since we don't have to discover the molecule, that stage is eliminated. We directly go from development to preclinical studies in some cases, but in the US and other markets, preclinical studies are not mandatory. Then we go for phase 1, 2, and abbreviated phase 3 trials. So instead of a timeline that spans over 12 to 15 years for a novel product, you can commercialize a biosimilar in roughly 6 to 8 years. Initially, when I started at Dr. Reddy's labs, they had commercialized the first biosimilar monoclonal antibody called rituximab, and that took them just a little over 3 years to commercialize. But at that time, the regulatory pathway was quite limited in India, and now with the advent of new companies, the regulatory pathway has become more and more complex, and they are requesting multiple studies for us to do. So that increases the timeline, which adds to the cost of the product, which adds to the R&D cost of the product, and ultimately it delays the patient getting the product. So these are some of the challenges that we have. If there are questions, please feel free to ask them. So this is the terminology that I used or that are used by the experts in the field. So the first one is called as a target product profile. That is also called as TPP. What is the meaning of this? So target product profile means you take the product that you're looking at, you buy multiple lots from the innovator, and then you run them through a battery of tests, all different tests possible, and then you come up with a picture of the product that tells you, okay, these are the different quality attributes of the product, or what are called as critical quality attributes, CQA. So you build a list of them, and then you take this and you say, okay, out of all these different quality attributes, what are going to be the critical ones for product safety and efficacy? And then based on that, you get some ranges of those attributes based on the innovator's different lots. And then you say, okay, this is what I'm going to manufacture. Now remember, each clone is going to be different from the other clone, right? So in some cases, you may not be able to manufacture the product exactly like the innovator. So you have to take a risk-based approach. So you have to say, okay, if I have certain differences in this quality area, will my product still be as will the product still have the same quality? That is, will it still have the same efficacy and safety that I'm looking for, or not? And so that is based on a risk-based approach. So you take a risk-based approach. You say, okay, this quality criteria will not be important in clinic, or this quality criteria is going to be critical for clinic. So based on that, you say, okay, if this is going to be critical for the patient, then I cannot vary too much. But in some other cases, you can say this quality attribute is not critical. Or rather than clinic, you can also look at stability of the product. So you can say, okay, because of this particular variant, high quantity of this particular variant, my product is going to be less stable. So that is why you have to remove some of it. So my point is that when you build a target product profile, you are going to sacrifice yield for achieving that product profile, because you will have to remove some of your product. So once you sacrifice yield, that leads hits the economics of your product. Now, since labor and other costs are cheaper in India, we can balance some of it out. But capital costs are much more expensive in India compared to the US because we have to import all the critical equipment. Media also has to be imported. So, all in all, it is not much more favorable for us to make the product compared to the innovators. Remember, the innovators have been manufacturing these products for the last 10, 15, 20 years. And they have had time to optimize the product life cycle. Many of them make their own media and they operate at huge capacities. So their cost of production is very, very low. So in turn, their profit margins are very high. And they use those profit margins to block us from entering into the market area. So we have to come up with innovative ways of making the product and shortening the life cycle, getting into market early, and hopefully to serve the Indian patients more than compared to the rest of the world. So then you have something called as critical quality attribute. So this is a quality attribute for which deviations from the safe. So if you set a limit for these attributes, and if there are significant deviations from these limits, then it can impact the quality, safety, or efficacy of the product. So these are critical and we cannot deviate too much. So then there are some things which are called as critical parameters. Critical parameters are the when I say parameters, parameters are physical levers that you influence, such as cell culture conditions or column operating conditions or buffer and media components. So these are the parameters that you influence, and those parameters which are critical for maintaining the quality of the product are called as critical parameters. So these quality critical parameters have to be extremely well controlled. And in some cases, depending on the severity of the parameter, you may have to have multiple or orthogonal layers of controls so that you ensure low risk of failure. So what does that mean? So, for example, virus removal. Now, your product has to be free of viruses, right? Adventitious viruses and endogenous viruses. Because cell culture products tend to have a lot of viruses, which are usually they will have no impact on humans, but you may be introducing some viruses through the cell culture conditions or during the operations, which are called as adventitious viruses. So the WHO mandates that you can have no more than one viral particle per million doses. So that's a very high standard of stringency. So in order to meet that, you cannot say, oh, this one step is guaranteed to meet that critical quality attribute. You will have to have multiple things and you cannot use the same. So let's say you're using a filter to remove viruses. You cannot use a filter multiple times and say, okay, I use the filter one time, it has given me 3 logs of reduction. If I use it 2 times, I'll get another 6 logs. No, because if it is used once, then it is used once. Then you have to have a different way of removing the viruses. So in some cases, you can say, okay, the column chromatography itself will remove some viruses. Okay, so you add that up. Then you say uh, some methods of inactivation, such as UV light or low pH inactivation. So these can also inactivate viruses, or temperature, high temperature can also inactivate viruses. So if you have multiple of them, you can combine the log reduction of each of them and say, okay, overall, my process is going to take care of 12 logs of viruses. And that comes to close to your safety margin. So for each attribute that you're looking at, you will have some critical parameters that have to be very, very well controlled. So these are your, and then there are key parameters. Key parameters are those where it will not impact the quality of the product, but it may impact the yield. Now, yield is very important for us from the industrial point because that is how the economics of the situation are measured. So you cannot have widely varying yield because you may have a certain loading capacity for your columns. And if you have too little yield, then it may be underloading the column. If you have too much, you are overloading the column. So that leads to uncertainty in the shop floor and there are difficulties in the manufacturing process. But it may not impact the quality at all. So those are just some of the parameters that influence the process, but they have no impact on quality. These are only related to the manufacturability of the product. So what are your input parameters? Input parameters are factors that you control within set ranges before the start of a process, such as before starting the process, you may do some load conditioning so that your load to a column you may dilute or increase the pH or lower the pH or increase or decrease conductivity or some of the buffers that you use. So you may have to control the pH conductivity of those. So those are the input parameters that you control before the start of the process. Then process parameters are those that are controlled during the operation of the process, so, such as residence time on a column or pressure buildup on the column or your oxygen cascade in the bioreactor. So where you have to monitor the bioreactor process over a period of days to weeks. And so you have to control the oxygen during that time, control the glucose during that time, control your media components during that time. So those are the process parameters. Then there are some things which are called as gaps. Now, before you start manufacturing and commercializing the product, you cannot identify everything in the process. You will focus on the top 90%, top item that affect 90% of your process. So the 90-10 rule, so called, or 80-20 rule. Because if you try to understand everything about every parameter before you start the process, then you will never uh, achieve it, because you will always be trying to understand something and something new always comes up. So you, as I remember, I said there's a risk-based approach. So you take a risk-based approach from the beginning and you say, okay, these are the things or parameters or attributes that I want to focus on. And this is where I'm going to spend most of my time and energy doing. And the remaining things will come over time. So what guidelines do you use for product development? Most of these guidelines are in the public domain, published by the FDA, WHO, International Council for Harmonization, ICH guidelines, and DCGI, which are Indian Drug Control Authority. They have Schedule M and Schedule Y, uh, and other country-specific guidelines are also available. So our overall approach is called as QBD, which is quality by design. So you cannot, the approach traditionally says you cannot test for quality in the end product. It has to be quality throughout the process. You cannot say that, okay, I'm going to remove bacteria at the end of it, so let the bacteria grow throughout the process. No, because that may lead to uncertainty. You don't know what all factors the bacteria are secreting during that time, right? Contaminants. So you cannot say, oh, at the end, I'm going to filter it, so there will be no bacteria at the end of it, and it is going to be safe. No, that is not the way. You have to design it beforehand. Or you have to say, okay, I can only have so much variance in the process. So they have to be controlled beforehand. So you have to optimize the cell culture conditions so that you have less of those variants and you control them throughout the process. You cannot say, okay, this one step is sufficient to remove everything. So these are the different ways you inbuild quality in your product and quality in your process. Because for biological products, process equal to product. The process is as important as your final product. So this is the management team for your product development. From the process development side, you have to have a cell culture process development team. The responsibility of this function is to develop and transfer to production a scalable process. Purification process development, their process is to develop a purification process and deliver it to the um manufacturing team. And it has to be a scalable process, because naturally at a small scale in the laboratory, you cannot do like 1,000 liter process, right? So you have to do it at 1 liter, 5 liters, 10 liters, and show that the same parameters are applicable. And then that shows you have reasonable confidence that even when you do it at 1,000 liters, it is still going to work. And then analytical methods. Here, because the product, all products are different from the previous products that you have worked with, you have to develop new methods to test for the product. Some methods are very standard, such as microbiology, sterility, bacterial endotoxin. So those are standard tests, but some of them are not standard, and these vary from product to product. which we will go through later on. So for product development from operation side, you have to have a manufacturing support group. This is the group that takes the process from R&D and transfers it to the shop floor. So their main purpose is to do the technology transfer. So they will be advising the R&D team that, okay, is your process scalable to go on the shop floor or not? For example, we had one process where they were doing refolding at 2 to 8 degrees, and that too for 36 hours. We said that is not possible because uh, 38 degrees, 2 to 8 degrees is not possible to be maintained on the shop floor because you will need a glycol chiller for that. And 36 hours is too long a period for us to block the facility. So you need to optimize the process. Because what happens in R&D, they do refolding in the cold room, right? So they have access to a 2 to 8 degree cold room, and then they leave it overnight or for 2 days because that is the way R&D people operate. But that is not going to be feasible for a commercial process. So this is where you have to have a balance between the R&D and the commercial process. So if you say no, I need a 2 to 8 degrees, which is critical for me. I cannot do it at any other temperature because I've tried the process at different temperature. It doesn't work. So when once you show the criticality of the process, then we may have to come up with some novel solutions. But otherwise, generally we tend to develop processes which are going to be feasible in the operations. Uh, then your production team, QC team, QA team. QA team is the overall responsible for the product because they will be certifying the product quality and the integrity of the process, and then only the product can go to the market and to the patients. You also need critical support from the facilities and the engineering groups. These groups will help to ensure that all your equipment is functional, facilities are functional, and they are being maintained in a timely manner. So what do you do? How do you approach the product development? First, you take your reference medicinal product, which is the RMP. Okay, so let's say you want to develop a biosimilar product of rituximab. So you'll buy some lots of rituximab from the market and you study them. Then you develop a target product profile. Then you do exhaustive physicochemical characterization of the product, which is you look at the primary sequence, you look at the secondary sequence, then you look at the primary, secondary structure, 3D structure, the stability of your clone, what are the different glycosylations in the clone, what are the different charge variants in the clone, if there are any lysine variants, all of that has to be looked at. And then you find the levers which are product responsible for product quality. So levers is something which I will talk to you in more depth going on, but you cannot have the same approach to all products, right? So you have to find out what are the levers that will impact this particular process. So in some cases, pH and conductivity may not matter. Let's say for protein A, when you're doing the initial purification, there your load pH is very wide. You can have a load from pH of let's say 6.5 all the way to up to 9 pH, and it will not impact the binding of the protein on the column. But for some other products, such as darbepoetin, your pH and conductivity may be very critical. So you have to find out where are the levers and for what purpose you're going to modify those levers to impact on your product quality. And then, of course, you have to have your process control strategy. Okay, how will I control my process? If I'm saying this lever is critical, let's say buffer and pH and conductivity is critical, then how do I maintain it? And let us say that buffer is going to be made at a certain pH and conductivity. Can I make the buffer at large scale also? When I make it at large scale, will it be behaving the same way? So, as an example of a bad process development, There was another group which developed a process for bevacizumab, and there they were going to do ion exchange for removal of some variants. The only way they were doing it was using a phosphate buffer. Now, the phosphate buffer, because they were using in the lab, they were using AR grade, but in the shop floor, they are using GMP grade material. So, first of all, the grades are different. As you very well know, grades will have the buffer quality, the chemical quality grades are very critical for the process, right? So they were using a different grade, and that too, there was a lot of variability between different lots of that product. So the way they were doing in the R&D lab was they would make a certain amount of buffer and do a trial run with it. And if that buffer worked, then they would certify that lot as suitable for purification. Now that is okay for R&D scale where you are doing only 5 ml column or 10 ml column. But if you have to do a 50-liter column, then making 35 CVs of that buffer, which is 50 into 35, like 1500 liters of that buffer, just to test whether it is going to work or not, is not a practical solution, right? But that was the mentality of those people who had developed it because they had not gone to the shop floor to work anytime. They said, this is the way we'll do it. But that doesn't work. So you need to have a very critical process control strategy. And you need to know, okay, this is what is going to work. This is not practical to be implemented. So during your development, you will first set up draft specifications. Then you will modify them as you get more knowledge about your product and what is possible in manufacturing. Your lot release assays, which is your how do you release the product to the commercial or market. And then you need to have ways to characterize your product physicochemically, and you need to do a stability testing of the product. So the purification process group will be responsible for developing a scalable purification process, and then they have to transfer the product process to the technology transfer team. So then they have to develop the draft BPRs, standard test protocols, draft MFR, manufacturing formulary record, and the draft IPQC, which is in process quality control scheme, and all of that. And that has to be going to uh, the manufacturing. This is a exhaustive overview of the product development approach in a functional sense. So on your left side, you have a process and analytical development which aims to achieve biosimilarity. So there are product related aspects which which are critical, such as some of them are glycosylation, then aggregation, then breakdown products, and these are all related to the product. So these are all influenced by the cell culture process, the purification department, and the analytical department. Because analytical department will test the product. They will need to know the limits of their testing. Okay, can how low of an impurity can they detect? Cell culture is responsible for the process. They are making the product. So they'll have to say, okay, these are the limits. I can influence glycosylation to a certain extent. I can influence aggregation to a certain extent. Whereas purification people will say, okay, I can remove the aggregates from the process. I can remove the breakdown products from the process. So these are part of the process related to the product quality. Then there are process related impurities, which come due to the process. So, for example, when you do cell culture, you may be introducing insulin in the cell culture to help the cells take up glucose. But insulin cannot be in your final product, so you have to remove it. So that is coming from the process. Then you have your columns. The protein is going through columns. So columns can leach out materials or break down products such as protein leachate. So that also cannot be in your final product. I mean, it should not be ideally, but there are always going to be a limit to that. So some amount of product, these process related impurities will be in your product. So you need to control them. And that is where you will put some limits on them. Some of these limits are established by WHO, such as host cell DNA. Whenever you grow cells, they will lyse and release some DNA. But initially it was supposed to be in the picogram range. Then the WHO modified that and said now you can have it in the nanogram range. Then you have viruses are part of the process, so you need to remove them. then you have the stability aspects, and you have something called as process and analytical tools. So these are the tools which will tell you if something is going wrong in the process. So if you expect the cells to have a viability of over 80% for 10 days, and if you see that the viability is crashing in the fifth day itself, you know something went wrong. So you can abort the batch or stop the batch or uh, immediately harvest it and go for purification. And these process analytical tools help you to set the specification. Then over the lifetime of the product, you will have what is called as process and analytical optimization. So what you will do is you will aim to improve the productivity of a process over time. You want to increase yields, you want to have a better product over time, and you may be even manufacturing in multiple sites. So typically most companies have a manufacturing site in US and in Europe as well. So they will have to have multiple sites of manufacturing, and that can lead to its own complications. So you will have to have a team for doing all of that. And over time, you have to file regulatory submissions with the authorities saying that your process is behaving as expected. You have to maintain a yearly trend analysis. Okay. And you say, okay, these are the 5 parameters that I'm going to be looking at as part of the product stability, as part of the process stability. So all of these has to be submitted to the uh, drug authorities in a timely manner. And if there are changes in your process or product, then you have to inform them. And then they take a risk-based approach and say, okay, these are not required to be worried about. So you can directly go ahead and implement the change. Or some of them may be very severe and they say, no, this change is very critical. You need to do a clinical trial again. So you have regulatory submissions which will vary from very simple to very complicated. So for all these, you need a team which will handle these and at different stages. So just to give an example, this is one of the maps, and we check the variants which are called as the acidic and basic variant. ACV relates to acidic, and K1 and K0 are the lysine variants. So the main peak or K0 is the majority of the product. So when you run it on a cation exchange HPLC, weak cation exchange HPLC, they will partition all these different variants into different species, and then you identify each species and you build a picture of the product. So now looking at the CX profile on the left side, that is a reference molecule. So you can see it has different acidic variants and different lysine variants, and this is a fingerprint of that molecule. On the right side is the size exclusion HPLC. So that tells you that most of the product is as a monomer and there are no aggregates in the product. So whenever you are making a product which is biosimilar, you have to follow this particular guideline. What it means is that you should have no new species in your product compared to innovator, and you should maintain the rank order of the species. So for example, your main peak cannot be less. So let's say here the main peak is the majority of the product in the RMP. In your own product, the main peak cannot be a small part of the product. Okay, even though you may have a similar profile of acidic and basic variants, but the rank order should be maintained. So peak number one should always be main peak, and it should be the majority of the product. You may have variation in different peaks, but it cannot be that your acidic peak becomes more than your main peak. So that is the meaning of that. But then again, you can take a risk-based approach and you can say that these acidic variants have no impact on the product quality, so I can include them in my product. Or you may find out that your acidic variants are making the product unstable, so you may have to remove all of that. So what happens is when you are removing an acidic variant or some variants from a product, you are also going to remove some of your main peak. So that will lead to overall loss in your process. So that is where you have to have a risk-based approach. Okay, can I manufacture product at a 50% loss of product during the process, or uh, is it safe for me to give a product with the higher percentage of variant? So that is the discussion that you need to have with the management team, and then you come up with your final product and process specifications. So for monoclonal antibodies, generally we follow templated process, which is basically you do the harvest of the culture, then you clarify the culture to remove all the cells. Then you do a protein A step, followed by a low pH hold, because the proteins come off the column in a low pH. So you hold them at low pH, that inactivates viruses. Then you can go to a second chromatography step, intermediate purification, such as cation or anion exchange. And then you go to final chromatography step, which is your polishing step. Uh, so in the cation exchange chromatography, you typically will remove some of the aggregates and charge variants. In the anion exchange step, you typically remove most of the process-related impurities, such as aggregates, not aggregates, sorry, such as host cell DNA, some viruses, and host cell DNA and host cell protein. After that, you go for nanofiltration, which is the virus filtration to remove any viruses, followed by your UFDF step, which is the ultrafiltration diafiltration step, where you do the buffer conditioning and concentrate the product down to your required specification. Because nowadays, and previously the products were all highly concentrated, like at least 10 to 25 milligrams per ml. Nowadays, they are making them super concentrated at up to 150 milligram per ml. So this step is very critical for maintaining that kind of concentration. But remember, during concentration, you tend to form more aggregates because all these molecules are coming together. Then you go for formulation and filling, that which is your final product. So this is just a stepwise manner of the process development studies. So what are the levers affecting your process development? So levers affecting purification process, as an example, is resin. Uh, for darbepoetin product, we filed a patent and it has been granted also. So what was unique about that process was that we used the same cation exchange chemistry 3 times in the process in 3 different columns. Okay. All of them were carboxymethyl cellulose, CM based. In 2 steps, we use the same resin, same uh, matrix also, but we use different pH and conductivity for that. And both were used for achieving different purposes. And in the same process, we use the same buffer pH and conductivity, but the resin matrix was different. So in one case, it was a saccharose base, and in one case, it was a different polymer base. So even though we use the same buffer pH and conductivity, we use 2 different matrices. So that actually helped us to achieve finer separation and removal of some contaminants. So this was unique to darbepoetin. Of course, monoclonal antibodies don't have that much of a complexity, but just to tell you how a resin can impact your process, and even the same resin using different backbones and different buffers can give you different outputs. Then, of course, residence time. How long is your protein going to be flowing through the column for you to bind everything to the column? How long you are going to wash it for to remove any impurities, etc. Then you need to do a load preparation. So one other team which was working on a protein, when I joined there, they were doing a, after clarification, they were doing a TFF step to reduce conductivity. So that was adding another 8 hours of time for the process, and that leads to instability of the product also, because the product is at room temperature for another 8 to 10 hours while you concentrate. Then I said, why do you want to spend time in doing the concentration? Directly dilute the protein with water and load it on the column. So finally, that is what we did. But the concern was, okay, it's going to be 200 liter, which will become 400 liter. I said, yeah, but you do an online mixing and it can go on the column. So that is where a load preparation is very critical. Then you have something called as intermediate washes, where you remove impurities. So you have to optimize those. Then how are you going to elute the protein? So it's not a simple thing, because let's say rituximab is formulated in a citrate buffer, but some other mAbs are formulated in acetate buffer. So how you elute the protein from the column is critical, because if it is formulated in a citrate buffer, but you're eluting in an acetate buffer, then you need to ensure that all the acetate ions are replaced by citrate at the end of the process. So that is another check you have to do. So you may want to minimize that ahead of time. And how are you going to elute the protein? Sometimes what happens is with MABs, aggregate comes off in the initial part of the elution. So you need to you need to collect it in fractions to remove aggregate. And I'll take you over a cation exchange step later on, which will illustrate this point even further. Of course, your hold time is very critical, because in the lab, you can keep the protein at 2 to 8 degrees or in a fridge and go home over in the evening. But in the manufacturing, you cannot do it. And many times there are power failures or equipment failures. So you need to be able to hold the product at a certain step. So hold time is very critical. So how long are you going to able to hold your product or process? Sometimes, let's say you load the protein on the column, but your next buffer is not ready because there was a problem in the water line or something. So how safe is it to hold the protein on the column? Will it get degraded, or will some contamination grow on it? So all of these are points that you need to focus on. These are going to be the levers where, if you develop the process well enough in advance, then manufacturing has less things to worry about. And exceptions. Okay, what are the exceptions that can occur? So just as I said, water may be an issue, or power may go off, or there may be exception where sometimes you may have more aggregate in your product for some reason. So all of those have to be noted down and uh, documented as much as possible. So this is a unit operation description. So every process is called as a unit operation, and like one chromatography will be one unit operation, clarification is a unit operation, uh, diafiltration is a unit operation. So we call it as a unit operation to make it simple for the manufacturing, because you can have one batch record for that. So what you have as a process description summary, you will have a process description of your process. How is the process operating? What is the process flow? What are your inputs, which will include your ranges and justification for that. Uh, After inputs such as which buffer, which resin, what is the bed height that you're going to use. Then you have your process parameters such as residence time, column volume. I mean, how many volumes of buffer are you going to pass through it? How are you going to elute the protein? Then you have to clean the column or the equipment that you're using. Let's say it's a bioreactor. So after you harvest the cells, you have to decontaminate the bioreactor, kill all the remaining organisms in that. Then you have to wash it and make it ready for the next cycle. Same thing for columns. You have to do a sanitization where you kill the contaminants. Then you have to do a cleaning of your column and have it ready for the next step, repetition of the step. And what are the sampling you are going to do? So in cell culture, you'll be sampling the cells every day to measure the glucose consumption, pH conductivity. Uh, so where all you are going to sample. And then you will describe what are your outputs in the process. What is your process output, which means, okay, I need to have the protein out at a certain pH and conductivity. The protein has to be in this volume. So parameters such as that. And analytical output. So you will say, okay, this step is meant for reduction of aggregates. So output aggregate should be less than 5% or 2% or 1%. And if this step is designed for removal of certain components, then you have to measure the removal of those components. Then you will describe what are the critical parameters, what are the key parameters, your whole time studies, your media lifetime studies if possible. Because everything that you do in development, and if you document that, that is useful for manufacturing. Because if they have any process deviations, they can use these to justify how they will approach those deviations. So BOM and COGS. BOM means bill of materials. So exactly what are you going to require for the stage? So let's say you're going to do a upstream process. So you will say, I need to, uh, so you will be issuing these from the warehouse. So you will say, I need 10 kilogram of cell culture powder, and then I'll need 5 kilograms of feed material. So, and you'll need these on a certain day and a certain time. So you need to give those to the issuing warehouse ahead of time, so that they will dispense the product and keep it ready for you in a double bag. So you take the bag, remove the outer bag when you go in your facility, and you use it. And COGS is also important because COGS relates to cost of goods, which is all the total cost involved in the product, which includes in COGS mainly we don't talk about manpower and utility cost and facility cost, but you talk about the chemical costs. What are the different raw materials being used for it? So if it is a media, how much is the media costing you per liter? If it's a resin, what is the cost of resin per liter? For example, protein A resins cost upward of $10,000 per liter. And if you only use them for 5 or 10 cycles, then it becomes very expensive. But if you use them for 150 cycles, then the cost come down. That is where the media lifetime becomes very important. So you want to be able to say, okay, I can use this media without any issue for 100 cycles, or I can use this media resin without any issue for 150 cycles. So that is where uh, the R&D development scientist also has to pay an attention to cost. It's not like you develop process and then you publish the paper with few sets of results and that is done. So just to give you an example, when my first paper was published, one of the comments I got from the reviewer was that n doesn't seem to be equal to 3. So I actually had done that experiment only one time, and that that passed the criteria, and it supported our hypothesis. But that was the comment I got. And typically for R&D papers or journals, they don't insist on a lot of repetitions. But if you go over to manufacturing, where you're going to take thousands of batches over the lifetime, then your N does become significant, and you need to show that your process doesn't vary a lot during that time. So just one case study, this is for protein A. So resin is I highlighted in yellow, but it is not a. In this case, it was a critical resin because the bead size was important. Uh, for this Procep VA Ultra, the bead size is large, so your flow rate is very high for this. So that is why I marked it in yellow. So in this case, you have inputs which go from equilibration of your column, loading of the column, washing the unbound materials off, and removing intermediates, which is the high salt wash that you're giving, and then the low pH wash to bring down the pH a little bit, and then you elute your protein. And then you go for column regeneration again. So on the left side are all the inputs that are mentioned. So for example, the equilibration buffer is 50 millimolar tris with NaCl and EDTA. Then triple CB refers to cell culture clarified broth, which is the um, broth that is having a protein. Then that goes for loading, and you say that culture volume, because the uh, titer of that protein was not very high, so we had to load nearly 35 column volumes of the broth on the column. And here you say, because I'm using a ultra resin, still it is a key parameter because it doesn't affect purity at all. It only affects the binding rate. So the residence time is very short, 1.5 minutes. So it's a very fast step because you're loading a lot of volume on the column. So you want to do it in a minimum time. Then you do a post-load wash to flush out everything from the system. Again, that is the initial buffer. Then you do an intermediate salt with the 1 molar NaCl so that anything that is uh, an impurity will come out of the column. Then you give an intermediate wash, which is slightly lower pH. But now, see, initially I had a background of tris and NaCl, but I want to elute it in citrate. So I want to remove all the tris from the column. So I now give you a wash with a 50 millimolar citrate buffer, but it is not at the low pH, it is intermediate pH. So this also removes impurities and it conditions the buffer. So you are removing all the tris and NaCl at the same time. Then you give the actual elution with the 50 millimolar citrate buffer at pH of 2.8. which is your elution. Then you regenerate the column with your orthophosphoric acid, which will clean up the column. So with this, I could do the same column for 5 cycles. So that what happens in manufacturing, you don't want to go for a very large protein A column because it becomes very expensive. So this is where the whole time also comes in. So instead of doing everything on a very large column, you take 1/5 the column size and you do the same loading and elution 5 times on that column. And because it's a very quick process, so it gets over within 1.5 hours. You can do like 5 columns in a unit operation, which is up to 8 hours totally, excluding the final cleaning step. So that uh, your protein is at room temperature in the whole condition only for 8 hours, but you maximize the use of your column and minimize your capital cost because the protein A resin is expensive. So this is what you optimize in your process step, and the output is called as protein A elevate. So what are your parameters that you are using here? On the left side are parameters, which is the resin, which is protein A, which is critical because without protein A, you cannot have achieve your purity, right? How do you control it? You control because is your protein A. What if you are going to somebody gave you a Q cephalose resin, right? So how do you control it? By your batch record. So in the batch record, it will be mentioned this is the resin to be used, and you compare the label on your column to the label on your batch record. That is your control strategy. What are the justification of this? Because you want to use protein A to reduce process related impurities or to purify the product, right? So that is why it is called as critical. Now, what resin am I using? I'm using Procep VA Ultra. It is a key parameter, not a critical parameter. Because during the preclinical trials, we have used this resin, and because it gives increased productivity, that is why it is good for manufacturing. That is our strategy for that. Then why you are using this equilibration buffer? Why is it critical? Because you are adding EDTA so that you remove any endotoxin in the product, and if there are any other DNA or other impurities, that will also be chelated to EDTA and not bind to the column. So like this, I've listed out all the different parameters that occur throughout the process, and we give justifications for those. We give a target for those parameters. Okay, pH conductivity should be 7.3 plus or minus 0.2, but you can have a range above or below that also. Uh, let's say conductivity, also you're giving a range because there is no justification for that other than this is what we use for our consistency batches. And NTB means not tested beyond these ranges. So we didn't have time to test all the different, like whether it will work at 30 millisiemen conductivity or not, whether it will work at 10 millisiemen or not. Because as I told you in the beginning, you cannot focus on all the parameters at the same time. So some of them which are not critical or key at all, we don't focus on those. But those which are critical, we have to uh, focus very strongly on. So, for example, your intermediate wash, you say salt concentration is critical because you are using it to remove impurities, right? So, that is how you will control it by using a batch process. And in the batch process record, when you add high salt to the column in your output, you will see a rise in conductivity. So, that tells you, okay, I'm actually passing 1 molar NaCl through the column. So, these are your load details, which is separate from your buffers. So the broth that comes out has to have a load, has to be 0.2 micron filtered. So you don't want any bioburden in that. What is the turbidity allowed for the load? Um, NTU stands for nephelometric turbidity units. So all of these are key parameters, not critical, because you don't want it to, if it is highly turbid, then it will choke your column, right? So you want to have and it shows you how well the clarification works. How much are you going to load? Usually you load 25 CV, but sometimes you may have less volume, so you may load only 20 CV. Sometimes you may have higher volume, so you may load 35 CV. So That range is given here. What are your process parameters such as residence time? Okay, for each step, what is the residence time I'm following? How many column volumes of each buffer I'm passing through? So all of this is captured, and when you hand it over to manufacturing, they have these parameters. What are your outputs? So you say protein A, pH should be critical because this is going for low pH inactivation. So pH should be between 3 to 3.5, for example. Conductivity should be around 3 millisiemens. What is the turbidity expected? What is the absorbance of the protein? Because you want to make sure that your product came out, right? So absorbance at 280 is your indication of how much protein is in the product. Then you will also check for column asymmetry and elution peak width, because if your elution width becomes high, then your protein gets more dilute and it shows you that the column has not been cleaned properly or that the column is degrading. So all of these are based on R&D data, for example, and you're checking those with the ranges that have been set up with the manufacturer. So here what we said was we tracked these for 63 batches on each column that we use. So we actually had two columns running in the lab and we did 63 cycles on each one, and that is how we have come up with these data. What are analytical outputs? Uh, how much acidic variant is allowed, how much main peak should be there, how much basic variant is allowed, how much aggregate is to be allowed. And most of these are critical quality attributes. So because these are going to impact your patient safety and efficacy. How much host cell protein is allowed, how much host cell DNA is allowed, and how much bioburden is allowed. So here I said bioburden is not tested because in R&D lab you cannot do bioburden testing because everything is open and you will have a lot of contamination. So other than that, all other things can be tested in R&D. So this is your typical chromatogram for a protein A column. Initially, you have the column readiness where you do the washing and cleaning of the column. Then you equilibrate the column. Red line stands for conductivity. Blue line stands for UV. So you can see that when I give high salt wash, the conductivity rises, right? And when I'm loading the column, that is a broth going through, so that will naturally have absorbance. So you can see the big block where you're loading the protein on the column. Then you do a post-load wash where some impurities are coming out. And in the high salt wash, you can see a very tiny peak which represents some minor impurities. Of course, we did not characterize those because they are very small and we do not have time to characterize these. And then you start your elution. In the elution, you see everything is coming off as a very sharp peak in a very small volume. And then you go for regeneration. This is your typical protein achromatogram. Now, what we did was we did a multiple cycles, 5 cycles at the same time, one after the other, one after the other, one after the other. And all five were behaving almost identically to each other. And we loaded the same way and we got the same output. Now, using this, this is a good justification for us to give to manufacturing that you can do multiple cycles if you want, or if you feel that you want to do everything in one cycle, you can do in one cycle also. Then we did media lifetime studies for protein A. How are your protein process outputs? How many cycles we analyzed for each one? What is the maximum pressure you got in any cycle? What are column variation? Now, in another case study is achieving biosimilarity. So remember the first product I showed you, right? This is the ion exchange profile of that product. Uh, if I'm going over time, please let me know. Okay. But I thought I could share some of it with your audience. So A represents the reference medicinal product. This is the fingerprint of that. But when we made the product, our in-house protein had a lot of extra peaks. If you see BV, is all the extra peaks that is not found in innovator. All the other peaks are found. So we said this is not acceptable and we have to match the innovator. How do we match it? So the only way to match it is to do a cation exchange. Uh, we hope to mimic the HPLC results using the process scale column and remove the basic variants in that. Okay. Next. Okay. So how do we do it? So we did a process chromatogram and we optimized conditions for it on the cation exchange. And as you see, we didn't do gradient separation because it's very hard to standardize or optimize gradients in a manufacturing environment. Whereas if you give a buffer, then they'll be able to use a step gradient very well. So here what we did was we did the loading of the column and then we washed the column and then we eluted. And we gave two different buffers for elution and we collected fractions of the elution and then analyzed them for your peak profile. As you can see, your initial pool one that you get, the first 5 to 7% of that is rejected because that has mostly the acidic variants. Uh, then the main pool, which is part of the first, second part of the first elution and the main elution, that is pulled together because that achieves biosimilarity. And the reject pool, which comes after that main peak, and you can see the dip in the UV profile and then a slight increase in the UV profile later on, that is all rejected. So because all the basic variant that we don't want come in that peak. So this is the way we optimize the process so that in the manufacturing you say, okay, reject the first 5 to 7% of the collect fractions, reject the first 5 to 7% of the protein, pull the next 30-35% of the protein and reject the remaining 15-20% of the reject fractions. And that way you can come up with this a product which is similar to your target. And this is the scalability that we did. So we did the process scale in the column where we took the process from a 4 ml column to 800 column. And we overlaid those chromatograms. These are process chromatogram, not HPLC, okay? So you can see that all the process chromatograms are also, we can clearly stack on top of each other. That shows that the process is behaving the same way, right from 4 ml to 800 ml column. So we are fairly confident that when you go to an 8-liter column or 80-liter column in manufacturing, you will find the same profile. And you will have to, when you collect the fractions, you will have to reject them in the same way that we are rejecting now. And you will get exactly the same product that you're looking at. So on the left side is the cation exchange HPLC data, which shows your main peak is around 45% and standard deviation and the minimum and maximum 45 to 55%. And all the other basic variants are almost zero. And the same thing with the aggregates also. So just to end the discussion, I want to emphasize on documentation. Because in many audits, when we face with FDA or other people, they will point out process deviations. So how do you tackle the deviation? So you say, okay, certain similar things that happened in the R&D. This is how we solve the problem. So that is part of your dossier. So everything that you do in R&D, you need to document. Whether it failed, that is also okay, because failure is very important for our knowledge. Because that tells you, okay, you cannot operate at this range. For example, you cannot operate at pH 2 because that will fail your product. So that tells manufacturing that you cannot go below pH of 2.5, for example. So, or you cannot go beyond glucose of certain concentration. So, because it may fail, right? So, all failures have to be documented, and there is no shame in having failures because it is very, very useful for manufacturing. So, what are if you look at the left side, these are the module name in a CTD. CTD stands for Common Technical Document, which is a module number of 3.1 or up to 3.2. This is where all your process and manufacturability is described. And DCJ has also come to accept this module, this as your submission. Is the internet on?
Yes, sir.
Okay.
Working.
Okay. So your primary documents are what go into your submission. So that is where you'll put all your limits of your process, how you're doing what buffers, what other things you're using for your process, what resins you're using. And then you have a list of supporting documents, which are not shared in the technical document, but these are the supporting documents that you keep with you so that when they come for audit, when they ask you, okay, how do you come up with this particular buffer? So you have a document with you, you say, okay, these are the experiments I did to use this buffer. So these are the supporting documents that you use. And just the reason I'm putting all of these is because a dossier is a very huge document, and it is the responsibility or the burden of regulatory authority or the RA department in your company to actually submit it. So they have to track all of these documents, and all the way right from the RCGM filing up to the clinical department, all of these documents have to be maintained and kept in a secure area. All the minutes of meetings has to be documented. So it's not a trivial process. That is what I want to uh, explain to you. Then you have a document tree where you have, after supporting documents, then you have development reports. Because supporting documents are what you will share with the clinical people, the regulatory people when they come to audit. Development reports are the reports underlying those supporting documents. So that will have actual experimental results in them, and your lab notebooks, which you will not normally share with the uh, regulatory team. But these are the documents that you need to have. So in case there is a deviation in the process or some issue happens, you will go back and review these reports. Then you need to develop an experiment planning system where you say, okay, this is what I'm going to do, and this is what I did. Because many times, if you ask students, they say, okay, I'll do this experiment, but they don't write it down. So when they actually do the work, they may have done something else. So there is no track of it. So this way, like when I was doing my postdoc, my boss used to say, if you want to do a different experiment, that is okay, you can do it as long as you do what I say first. So we discuss, we agree, okay, this is what I'm going to do, but I may disagree with him. I may say, yes, I don't think that idea is very good, but I have to do it anyway because of the agreement. And then he says, you are free to do other experiments also. So that is what uh, we encourage. Like, we don't want you to just do experiments on your own. So what you will write down is, okay, this is what I'm going to do. So you write it down. And then after doing it, then you say, okay, this is actually what we did. Now, why are both of those important? Because when you say this is what I'm going to do, it may happen that at night the power went off or the reactor stopped stirring. So it didn't match what you intended to do. But at least you have a record of that. And over time, you will become better and better at actually doing what you said you were doing. And that is when the process becomes mature enough to transfer for a manufacturing department. Then you will have a hierarchy of documents. So overall hierarchy is what you will say, okay, I'm going to start work on this product called bevacizumab. Okay, so that is your initial highest level document. In that you say we will develop the critical quality attributes of this product, the target product profile, how you're going to clone the product, how you're going to purify or at least you will lay out a strategy for how you are going to purify the product, how you are going to or do the clinical trial, when are you going to do different regulatory submissions, how long is it going to take. So all of that will come in the master document. From that master document, now you have a basis to define initial documents. Okay, so you said we have to generate the clone. Are we going to buy a clone or are we going to make it in-house? If you're going to make it in-house, this is the way we are going to do. What is the process steps required for RCGM filing? Okay, these are what is required. So these are the tests that you will do. What are the process steps required for clinical filing? So this is what you will do. So all of this comes out of the initial strategy document. Then you obviously have templates such as what is the template used for chromatography process. So we have a uh, document called as Chrome template. So in that, what you do is you take a printout of that initially and write by hand. Okay, what resin is going to be used? What is the column volume? what are the different buffers that are going to be used, what is the residence time, what are the pressure limits that you're going to set. So you write it down in the initial document, and then you hand it over to your bench scientist. They will perform the experiment, and if there are any exceptions during that, they will make a note and they'll say this is what we did. And both those documents, along with the chromatogram, are printed and put in your lab notebook. So now that is your evidence. So over time, you build a whole hierarchy of documents based on a lab notebook. Then you summarize those into a report, and then the summary of summary is given as your regulatory submissions. So your system has to specify how you're going to review your documents, how you're going to number the documents, how you're going to track the version. Uh, Let us say you make the initial standard test protocol in 2014. Then you say this has to be periodically reviewed. Otherwise, you may carry on mistakes in that process. So you say every 2 years it will be reviewed. So how? So it means that if the document was made in 2014, it has to be reviewed in 2016. So your quality system people will come and say now this document has to be reviewed. So who are going to review it? And after review, you will say, okay, these are the changes we want to make, or there are no changes to be made. Now the version has to be changed because initially it was a 1.0 version. Now it has become 1.1. That shows that the document has been changed, at least the numbering has changed. So that is the different version from what we started with. So that gives you a way to track your different documents. And you have to have document numbering, because how do you know that this document belongs to this process of, let us say, protein A, protein bevacizumab versus trastuzumab. So your numbering has to be very consistent in that, right? You cannot take documents from one project and use for a different project. And then how do you organize them in your server? How do you archive the documents? Some of them have to be kept in an earthquake proof room or in a fireproof room. And all do you keep the archives? Do you back up on site or do you back up off site also? So all of these are part of your experiment planning system. And these are the things that you have to keep in mind. So we have come to the end of the talk. Please feel free to ask me questions.
Uh, Participants, if you have any questions, please tell me. Otherwise, I received some questions, sir. I will ask. Okay, I think there is no questions, sir. Just I am asking, sir, one question. You have suggested many types of evaluation technique, biosimilars products. Actually, what is the life cycle of the biosimilars evaluation? Exact cycle of biosimilars, life cycle.
So first is in biosimilars, uh, there's actually a very good diagram in the ICH Q5, I believe. So you start with the initiation of product, where you develop the product and do the testing. Then you go in for different testings, clinical trials. Then you do commercial scale up and manufacturing. This is your one part of the life cycle. But during this, there is always going to be continuous improvement because you want to improve your product and process all the time. As you gain more knowledge of your product, you will say, okay, maybe I want to modify this process parameter. Maybe I want to modify this. Or it may be that country A requires a certain document. So you need to generate it in R&D and use it to support that filing. So lifetime improvement is continuous, but there is also called as planned obsolescence, which means that the product life is going to end at some point. So let me give you an example. For example, GCSF. GCSF is a very old and historical product, but then its replacement came out as a pegylated GCSF, right? So nowadays, the innovator has stopped making GCSF, and you are not going to be able to what do you call even find the innovator to do clinical trial. So you can only do a PEG GCSF. Similarly, for many drugs, such as trastuzumab, for example, it was first available as an IV formulation, intravenous. Now, company has modified that and gone into a subcutaneous formulation, so that it is a very small injection. A patient can take it at home also. So that shows, because they want to improve the use for patient, and they also want to keep some marketing for the innovator, right? So they will come up with new versions of that product. Similarly, for insulin, you have certain analogs. And for example, some others are going to be retired, like uh, liraglutide has come out in the market, but probably semaglutide will replace liraglutide over time. So you need to plan for that. So you need to know that the product will not be indefinitely available for me. Some products, yes, like rituximab, they are always going to be used for that indication. But then, uh, like non-Hodgkin's lymphoma, rituximab will always be used for it. But trastuzumab by itself is not very effective for breast cancer. What they're doing is coming up with antibody drug conjugates. So what they do is attach a radioactive molecule or some other molecule to the tail of trastuzumab and chemically conjugate it. So then when the antibody binds to its receptor cell, it actually goes inside, gets internalized and kills the cell. So you have newer, more efficacious versions of proteins. Then you have something which are called as biobetters. So if you know, okay, rituximab binds to CD20 molecule, then we have another molecule which, if you do a little modification, that will bind even more strongly to that CD20. So those are biobetters. Then you have novel delivery mechanisms. So insulin, a lot of push was trying to make insulin as a nasal formulation. So you can just inhale that instead of injecting. That of course did not work, but you have to be ready for that. And your manufacturing has to be flexible enough that you know, okay, in the next 5 years, this product will be outdated and we need to focus on the new one that is coming up. So that is part of the life cycle.
Definitely, sir. And, thank you very much, sir. First of all, I thanks our respected VC, sir, Professor K.R. Sambasivar, sir, who's invited to you for this a fantastic session, and who inspired us and encouraging to conduct this webinar and made us take it further. Sir. After that, we are grateful and would like to heartily thanks to our today guest and speaker, respected Dr. Darshan Koticha, sir. He is currently working as Director R&D Virchow Biotech Private Limited, Hyderabad, Telangana. It is a great privilege that the respected sir joined us today webinar, sir, who gave a fantastic and informative session and addressed on the specific topic approach toward biosimilar product development. Actually, the process of biosimilar medicine development involves rigorous analytical study to establish a comprehensive understanding of the similarity of biosimilar to the reference product, assessment of toxicity, and clinical studies are also used to further establish similarity. We thank you very much, sir. Thank you to all.
Most welcome. And it was my pleasure to talk to the students. And unfortunately, I had to compress everything into a very small talk. So if your students individually have questions, please feel free to approach us and come up with questions, and we can try to solve them. And if they want more hands-on experience, they can come and actually learn on the R&D floor with us.
Yes.
And we'll be happy to give them a practical understanding of the topic as well.
Thank you to absolutely. Thank you to all students of
Thank
pharmacy,
you, sir.
forensic,
Thank you.
and agriculture. Thank you,
Thank
sir.
you.
Again, very thank you.
Thank you.
Thank you, sir. Thank you.